Again, I am here with the request to focus on blood-spinal cord barrier, and I must tell that this is joint research work with various organizations. This is my Uppsala University. This is the Air Force Office of Research in the United States, European Office of Aerospace Research Development, National Center for Toxicological Research, United States, FDA, NIDA, National Institute of Drug Abuse. This is my hospital, just completed 300 years, and US Air Force Base Research, together with AstraZeneca as drug company. Actually, in medicine, sometimes what we treat is symptoms, and we do not go for the root cause. And that is the basic problem. If we go to the root cause to treat the disease, then symptoms will vanish anyway. So this is a very old saying from an Indian say. So we believe that in central nervous system, blood-brain barrier could be one of the root causes. So we are doing this work since 1977 at different places, you can see, and since 1988 we are continuing till date in Uppsala University. So rightly pointed out here that astrocytes play a very important role because glial cells are 10 times more in number than neurons, but endothelial cells are even more. So we should look non-neural cells, and astrocytes could be one, microglia are others, and of course our research is focused on endothelial cells to control the neuronal function. We summarized this book in 2003 and telling that the good thing for our part is drugs can influence this blood-spinal cord barrier or blood-brain barrier. And apart from that, there are also other barriers existing, that is blood CSF barrier, that is not in microvessels because they are fenestrated, but they are lying in the epithelial cells of the choroid plexus. There is also a barrier, a mild barrier between CSF and brain cells, but this is very less tight. So we have a barrier system. This cartoon explains clearly that blood-brain barrier is the most tight, followed by blood CSF barrier, and we can have a still exchange to something between brain cells and extracellular fluid compartment. However, here I must want to tell you that if we have a barrier between blood to brain, probably we have equal barrier from brain to blood. So many substances that are present in the brain, if you have a stress, many neurochemicals are released in the brain. Normally they are not going into this vascular compartment. If they go to the vascular compartment, you will have a rebound effect. It is still not known that whether in neurological diseases both barriers are equally disrupted. So this is one of the research that we are carrying out at the moment. And the most important part on the disease process in the central nervous system is stress. That can be at the cellular level, that can be at the molecular level, as my previous speaker has shown so many changes in gene expression, enzyme and pathways. So these are molecular stress. They can occur anywhere. You can also check in cell culture. Cell culture is one of the good ideas because you can study the pure effect. But you cannot understand the balance because of course in the body if you have injury there are so many things are connected. In cell culture you study only one thing. You give one thing, astrocytes react, neurons react. But therefore in vivo experiments are much more closer for the human experiments. And as I told you that these epithelial cells of choroid plexus are connected with tight junctions, the microvessels are fenestrated. So my point is that we still do not know whenever we have a breakdown of the blood-brain barrier, do we also have breakdown of the blood CSF barrier? Or which one comes first? This is the first example given by Milton Brightman at NIH Bethesda. This is the lanthanum that we are using. It is stopped between the two endothelial cells, and this opens the idea that the tight junctions are the important pathways for the blood-brain barrier. But apart from tight junctions, the cell membrane can also act as a potential barrier. And this is the study we did in Germany. So in our laboratory, this is a rat spinal cord, and we have injected lanthanum, and you can see that this is a normal microvessel, and they are not leaking into the endothelial cells. Tight junctions are intact, and this is one neuron, and the astrocytes are also present around. So neuropil is very compact here if the barrier is not leaking. But when after injury, in this case this is heat stress, you can see that the barrier is leaking to the same lanthanum, although the tight junctions are intact, but you can see that the endothelial cell infiltration here, even overlapping the tight junctions. So this is a new phenomena in hyperthermia, because you don't need tight junctions to be widened. You can still have increased permeability by damage to this membrane, and of course the tight junctions are also membrane. They can be damaged without widening. Rightly pointed out by you that in clinical cases, each case is very different. So it is very difficult to control. We cannot produce clinical cases in experimental situations, and we have no idea that what individual differences are. So these are very important. The same thing is also, we have different genetic factor, metabolic factor, and that's why nowadays US FDA is trying to create personalized medicine, and I think you are also very much aware about this new situation. So we started our work on a spinal cord injury in 1988. This is my colleague Thomas Winkler, and this is Professor Eric Stolber. The book we have written also 10 years later. So this was the book where we summarized electrical activities and spinal cord injury, contributed by 25 other experts. But still we do not know that in human cases we have less idea about drug interaction, disease interaction, because patient could have many diseases. Environmental interaction, I told you, nanoparticles or whatever, stressful interaction, and all other these factors all influence the final pathophysiology of any injury process, and therefore the treatment. Here in this institution, I did not tell that this is very common scene doctors or neurosurgeons have seen. In this case, this spinal cord is completely injured. This is one of the rare cases. Doesn't occur normally in that. So there are several models, animal models, and I told you earlier, we cannot establish any animal models that is very similar to clinical cases. We can't have it. So the transaction, crush, and contusion we did not like, because we should find a minimum model. Consider if the 20-gram weight is dropped on rat spinal cord, and consider to human, if it is transferred in terms, there are some papers and we also calculated, it amounts to 260 kilograms. This is quite unnatural. And in mouse, if it's 10 grams is done, then in human cases it is about 80 kilograms. So I mean, we cannot simulate human cases of the spinal cord injury in any way. So we wanted this mild trauma that acquires only 4% of the spinal cord in one side, and you have seen this slide. So, and this is the example to see that we have recorded spinal cord evoked potential. You can see that on the dorsal side, it immediately disappeared. But on the other hand, in the caudal side, it has increased. And since you people are here doing patch clamp and many other things, it is quite clear that this has increased amplitude because the loss of inhibition. But what you can see that in both the cases, there is an increase in latency here. But after 120 minutes, even after 10 minutes, there is tendency to recover. And this is positive injury potential, you can see very clearly. So under these circumstances, the spinal cord looks like this. This is control, and this is the experimental. You can still see that one side is much more swollen than the other side. So this is perfectly reproduced in our animal model. And when you see that the blood spinal cord barrier to Evans blue is a large molecule, and it combines with albumin, so it becomes about 68 kilodalton, and the leakage is tracer protein complex. Here the important point is that we have different segments, C5, T4, T9, T10, 11, T12 and L5. So before, above the lesion and below. You can see we can detect leakage after 1 hour in different segments, not in the far remote segments, and that is increasing over 12-hour period. The important point you can see here that there is always a difference between this is the injured rostral and caudal. They behave differently. But if you use this 131 iodine, you can see leakage can be seen in the injured area within 5 minutes. So this is the effect of different traces. You use different traces, you can have different results in the same model, and also the magnitude and intensity of increase, because 131 iodine binds to different proteins and it is smaller in size compared to advanced blue albumin complex. And this is an example here that this lanthanum is present in the basal lamina. But this slide is very important because you can see one endothelial cell is infiltrated with lanthanum, but the others are not. So in vivo situations, if one cerebral capillary or a spinal cord capillary has 2 or 3 endothelial cells, only a part of the endothelial cell membrane is permeable, not the whole. So even in vivo, these differences can count. So you can understand there is a lot of difference between cell culture and in vivo experiments. Because if this part of the endothelial cell that is not permeable were used, you can get different results. This is another example. Here you can see that lanthanum is leaky and the vessel is still round, and you can see that here edema and myelin vesiculation is clearly seen. But you can see that tight junction is really locked here. It is not open. Lanthanum is not going through this way, although we can see lanthanum in microvesicle profiles. So this suggests that blood-brain barrier or blood-spinal cord barrier is a key factor in causing other functions like edema. Whenever proteins are leaky, I mean you are very familiar, it produces edema. And here you can see that we can detect changes in water content as early as 5 minutes after injury. If you use electron microscopy, you can detect 2 minutes after injury. So this depends, and it is gradually increasing up to 12 hours, and still the rostral and caudal sites show quite remarkable differences. Neuronal changes, you can see here that this motor neuron is very dark, almost dying, and spongy neuropil as compared to the normal from the same segments taken. And this is T9 right side, it's not injured directly. Glial cells, this was very important and mentioned. You can see that this is GFAP, T9 right side and T9 left side. Both showed massive upregulation within 5 hours. So previously, people believe that gliosis can take several hours or days or weeks. This is wrong. It can occur even 2 or 3 hours. And these are drug sensitive. Of course, these are pre-treatments. Naloxone and endomethacin were given before making injury. And you can see there is no gliosis. So whenever there is an injury, both neurons, glials, and even myelin, that is the constitution of axons, can be seen here. This is myelin basic protein in control cases, and you can see here after traumatic injury, T12 right side, you can see the swelling of the dorsal horn and much less intense. All these fibers are gone, means axons are lost within 5 hours. At that time, people believe that pathology comes only weeks, months, and years. But this is not true. When we can detect physiological changes or activity within milliseconds, I mean cell changes can also occur. So this is an example of C4 expression in dorsal and ventral horn after 5 hours. We always believe in C and confirmation, so you can see why we have used this unilateral incision of the dorsal horn. Because this is T9 left and T9 right. You can see left using transmission electron microscopy, here are massive holes, edema, activated microglia, and there is one microvessel that is collapsed up to half, but lanthanum is here, leaky, and you can also see the myelin changes. The right side has much more myelin damage and open spaces than the left side. So whenever you make an injury at one side, even in the same segment, the structural changes are different. Then coming to treatment. These are the treatment. This is untreated. Then T9012. When we have given BDNF, we have applied topically because there are questions. At that time, BDNF was terribly expensive, and even nowadays. So if we give intravenously, it will be diluted. The amount that is reaching to the spinal cord is not known. So we have applied topically, and you can see here topical application has really produced nice results. But when we have given a mixture, BDNF and GDNF, then we can have even better result, because the spinal cord or brain requires neurotrophic factors both from brain derived, glia derived, and even I can show you later that other factors that are very important. And you can see here that this is the control spinal cord injury. The question was when and how we can apply the drug to have the neuroprotection. Here you can see minus 30 minutes is perfect. Even up to 10 minutes after injury is quite good. 30 minutes better, but 60 minutes after practically there are no changes. So the time point of topical application is very important. This is given IGF-1, insulin-like growth factor 1, in the identical doses, not in nanomole or something like that, not in equal molar doses. But here you can see that 30 minutes after IGF-1 in identical doses was much more effective than the BDNF. The same thing is happening to the leakage of radioiodine. BDNF up to 30 minutes is quite better, 10 minutes is the maximum. But here you can see that even 30 minutes is much better. What could be the basic mechanism of topically applied BDNF or IGF-1 on a spinal cord? At that time, it was quite not known the involvement of nitric oxide synthase. So these are nitric oxide synthase level neurons in 5 hours. You can see after BDNF, they were not only able to reduce, you can still see that there is some elongation about that, and this is from IGF-1, so at low magnification. So the point is that whenever you have, there is no known connection even nowadays except some connection with cytokines that BDNF can influence nitric oxide synthase application. But at that time, we have seen, this means that in vivo situation, there are crosstalks occurs in many other partners that are not known before. To test our hypothesis at microscopy, here you can see that this is the spinal cord injury untreated. This is volume of swelling, and this is small magnification to show you. You can see that motor neurons are well preserved. This is leakage of lanthanum, and in this case there is no leakage of lanthanum. It means if you have neuroprotection, there is no breakdown of the barrier to even a small molecule tracer like lanthanum. And of course we believe in multiple combination and physiological things. This is myelin basic protein in untreated group. You can see even one side is much more swollen and axons are lost. But when we have given theoretically nimodipine before treatment, of course, you can see that how much spinal cord and these myelin is well preserved. So these are experimental conditions. In such situation, also there is no leakage of blood in that. Then together with Professor Wang, we try to study a different combination of neurotrophic factors, and we study on another parameter, nitric oxide I have shown. There is also carbon monoxide that is called hemoxygenase 1 and 2. I briefly go through this. So you can see hemoxygenase 2, this is constitutive isoform. After a spinal cord injury, it has increased so much. And you can also see in different areas after trauma. When we give as a pre-treatment with a serotonin synthesis inhibitor, it is known to reduce breakdown of the barrier and also edema. You can see that this volume of spinal cord ventral horn is reduced and very less number of hemoxygenase are present. So whenever we have reduction in cell injury, the blood-brain barrier or spinal cord barrier is reduced, and these factors that are upregulated are also downregulated. So the question is, which combinations are good? So then we have used another factor, CMTF, BDNF and GDNF. Then we have different combinations, and we have given in this doses topical. And you can see here that is iodine extravasation. This is untreated. The best result we have got, this is 3 combination of the neurotrophic factors as compared to the any kind of 2 combinations. This is also occurring the same in water content. The 3 combinations are much more better. I do not have time to show you that if we change other neurotrophic factors, we could have cancellation of good effects. This is the spinal cord blood flow. This is quite good, and it is reduced after spinal cord injury, and the 3 combinations are still able to maintain a higher blood flow measured by microscopic techniques. Neuronal damage can also come here. 3 combinations are much better than any other 2 combinations as compared to the untreated spinal cord injury. HO2 expression also follows in this way, and you have seen this previous example, IGF-1 and how they have regulated HO1 expression, just very similar to nitric oxide, but they are not identical. And here are the example, if we have given a combination of all, the result is best as compared to this untreated group. So the different combination of neurotrophic factors are doing better. We have to find how many combinations are good, but if we include 1 or 2 other neurotrophic factors, the results are cancelled. The question comes, how near we are to the human cases, and are we doing entirely different or only theoretical basis? The answer is that this is an example of human cases with a spinal cord injury, relatively same area, and 6 hours after. We have done albumin immunohistochemistry. You can see that nicely a special pattern of albumin leakage can be seen in human cases. So this suggests that the opening of the barrier is also very wide in the human cases. But here is the problem. I don't still know that 6 hours of human life can be how many hours of rat's life, because rat lives only 2 minutes. These are hypothetical and other questions that I have no answer, but at the 6 hours, you have the results. Then again the question is, this is not a spinal cord injury. This is a spinal nerve lesion that causes neuropathic pain. And you can see also albumin level motor neurons after different areas of the ventral spinal cord. So it means that even long term, these are 6 weeks, I guess the neuropathic lesion can also open the barrier in the spinal cord. And I will only show you some example. I showed earlier that even silver intoxication itself leads to opening of the barrier. No other treatment, just silver treatment. Then we have given silica oxide as nanoparticles treatment in same spinal cord injury, and you can see that the silica treatment results in this spinal cord injury as compared to the untreated spinal cord. But the most interesting point is here that after silica, you see complete loss of motor neurons and sponginess as compared to the untreated. These are leakage of the blood-brain barrier. This is silica treatment. So silica treatment before and injury given after exacerbates spinal cord pathophysiology. So again we come to the same area that whatever CNS insults can be influenced by environmental, chemical, stress or whatsoever. It opens the barrier, causing edema, necrosis, and lead to apoptosis, gene expression. All together, the combination can lead to cell death. We can either protect here, we can either protect here, or even we can protect here. So what my previous speaker has shown that probably they are talking to prevent this aspect, and that's why they are having better results as well. So we believe that we have to keep an open eye and try to see results from different angles to reach a goal. We have different collaborators from various aspects of these are Swedish collaborators, American collaborators, and also we have some collaborators from different parts, United States, India, Australia. We have support from different organizations. I'm not going to read, I don't want to take your time in this case. And I just quote with this, that normally people feel that you are only responsible what you are doing, but it's very old saying that we are also equally responsible for those things that we have not done. So this is also our duty to understand the fundamental of medical treatment and its modulation by various diseases. So with these words, I'll stop here and look forward to listening. Thank you very much for the opportunity.
So thank you very much, Professor Sharma. So any questions for Professor Sharma? Yes?
Thanks a lot for your excellent lecture. My question is, as we know, NGF can improve the sensitive function, and BDNF can improve the motor function. But as you study only about the focus of the BDNF and the GDNF, can you give some opinion or suggestion?
We have studied also NGF and NT3, NT4. We have published this paper as a review in Current Pharmaceutical Design in 2007, telling a suitable mixture of or combination of neurotrophic factor can enhance neuroprotection. So in our hand, or in this model, when we combine NGF or NT3, it has cancelled some of the effect of BDNF. So we do not find that the effect is added. Of course, if we apply NGF alone, it has some improvement, but we do not understand its biological mechanism so far.
Thank you. And also I have the opinion, if you target or lesion the peripheral nerve, can secrete the high neurotrophin. Maybe in spinal cord, if injury, maybe also the neurotrophin can the secrete high level.
Yeah.
But do you know maybe the period after the 2 hour, 6 hour, and today for 7 days?
Yes, in peripheral neuropathy, neurotrophic factors are known treatment to reduce the morphological structure and even sensation of pain. But you know what happens that in neuropathic pain, the spinal cord pathology progresses, but you can have less pain symptoms. Your pain sensation can improve while the spinal cord is damaged. So therefore, we have not studied by ourselves in our laboratory on neurotrophic factors in neuropathic pain, but there are some reports, even in clinical cases, they use these neurotrophic factors to reduce neuropathic pain. But spinal cord pathology, to my knowledge, no one has studied so far, and we have no time to do that, but we have used cerebrolizin, that is a mixture of BDNF, GDNF, IGF-1, and also NGF and CNTF and some other active peptide factors. They are working to reduce the sensation of pain, and also in our hand, spinal cord pathology. But this is the area where more research is being done. Thank you for asking this question. Yes, please.
Thank you, Professor, for your wonderful lecture. You mentioned about the brown berry. It's especially on the hypothermia. Those kind of stress can influence brown berry. My question is about the surgical patients or anesthesiological patients have a lot of stress.
Yes.
What kind of influence on the BBB?
Yeah, this is a very good question, and I think in early '65 there was a book by Hans Selye. He wrote Stress in Health and Disease, and he mentioned some observations in Canada that surgical stress can also open the barrier, because this is also a stress, but it depends on how long and how intense it is.
I have a question.
Yes,
Did you introduce the markers, serum markers of the patients of the dysfunction of the brain blood barrier?